Okay, today we're going to move on to talking about DNA and how DNA gets copied, also known as replicated, before a cell divides. I want to go over a little bit of history, though, on how it was discovered that DNA was the genetic material or was the inherited material. So here's an interesting experiment that was done. And you don't need to write down like every little detail here. I'm going to tell you at the end like what to write down. But this is kind of cool. So this researcher, Frederick Griffith, in 1928, took some bacteria. So you see right here where my cursor is, this is bacteria. He injected the mouse with the bacteria and the mouse died. All right. Here is a different bacteria. We would call this one non-pathogenic because he injected it into the mouse and the mouse lived. So living S cells are pathogenic, mouse dies. Living R cells are non-pathogenic, the mouse is alive. In this experiment or this trial right here, he kills the pathogenic bacteria, injects that into the mouse, and the mouse lives. So basically he's taking this first one, killing it, injecting the dead bacteria cells into the mouse, and the mouse lives. This is the one that's the most interesting, though. He takes the dead S cells and the live R cells. So these two right here. So remember, each one of these, the mouse lived, the mouse lived. Now if he mixes them together, the mouse dies. So what happened? So basically, his conclusion is that there is a bacteria can transform into deadly bacteria with an unknown heritable substance. So next to his name, I would just write, there is an unknown heritable substance. That was his conclusion. There's something that gets passed from living things. And bacteria can actually do that. They can pass DNA from one to another, which is kind of scary because if there's a bacteria cell that has antibiotic resistance, it can pass the resistance to the bacteria, its neighbor. So that's kind of scary. So Frederick Griffith, there's this unknown heritable substance. Avery Oswald discovered that the transforming agent was DNA. So next to his name, that transforming agent that Frederick Griffith was talking about, the bacteria passed to the other one, was called DNA. Okay, these guys right here tried to figure out Hershey and Chase. So this was kind of what happened. Hershey and Chase wanted to confirm what Oswald Avery concluded. And so what they did is they took these viruses. Did you know that there's viruses can actually infect bacteria? They're called bacteriophages. So they took bacteriophages and made them infect. And so when a virus infects a bacteria, right, you can see right here, they land on it. So this crazy looking alien, I don't even know what that is, what that looks like. It's a bacteriophage, but it looks really crazy. What they do is they land on the surface of the bacteria and they inject DNA in, and then the bacteria makes more virus from that DNA and it like explodes the cell and virus is released. So yeah, a bacteria can get sick. So what they did was they radioactively labeled sulfur and radioactively labeled phosphorus. So if you remember our macromolecules, if radioactively labeled sulfur showed up in the bacteria, that means that they're injecting protein. So they didn't know what these viruses were actually injecting. So that means that they were injecting protein. If radioactively labeled phosphorus showed up in the bacteria cell, then they were injecting DNA. So they were just confirming a virus is going to inject the genetic material. Well, in this situation, is that genetic material protein or is it DNA? And so what they found out was the virus infects the bacteria, no sulfur shows up. So if it's pink, that means that it's there. No sulfur shows up. So therefore they can conclude The virus did not inject protein. In this one right here, what happens? Phosphorus showed up. The radioactively labeled phosphorus showed up inside the bacteria. So under Hershey and Chase, I would just put the conclusion, because this is a little bit higher level than what we need to talk about in this class. But I just wanted to show you exactly what they did because it's actually a really cool experiment. I would just write under Hershey and Chase, confirmed DNA is the genetic material. Confirmed DNA is the genetic material. Okay. Edwin Chargaff then came up with the base pairing rules. And I'm going to tell you what these bases are. So I don't know if you learned this in middle school at all, but DNA, the DNA code only consists of 4 different letters: A, T, C, and G. And I'm going to tell you what to write down under this guy's name, but I just kind of want you to pay attention to this because I am going to talk about it again. And I do have this picture in your notes. So all I want you to put under Edwin Chargaff is that even though the DNA composition varies between species, so DNA composition varies between species, but the amount of A equals the amount of T, and the amount of G equals the amount of C. So therefore, he came up with the base pairing rule. A pairs with T, G pairs with C. And so I know you may be kind of confused right now if you don't have a lot of background on DNA, but I'm going to show you the structure of DNA and this will make more sense. So DNA, remember, is double-stranded. On one side of the strand, if there's an A, then it's going to have a T matching up to it on the other side of the strand. And then on one side of the strand, if there's a G, it's going to have a C matching up to it. So that's what you need to write down under Chargaff. Chargaff's rule is just the base pairing rule. A goes with T, C goes with G. All right, Rosalind Franklin. All I want you to put under her name is she found, or she took the first image of DNA. Took the first image of DNA. That's all you need to put. She took the first image. Then Watson and Crick took her image and they discovered the double helix. So all I want you to write down under Watson and Crick is discovered double helix. So there's your history lesson for today. Thank goodness we know all this stuff now. Let's talk about the actual structure of DNA. So DNA is a double helix, meaning it's double-stranded and it twists around. The backbone of DNA consists of, so if you notice right here, DNA is made up of a bunch of nucleotides bonded together. Remember, a nucleotide is a phosphate, a sugar, and a nitrogenous base. And so the A, T, C, G, those are your nitrogenous bases, all right? So the backbone of the double helix is the sugar and the phosphate. And remember, our sugar is it's a 5-carbon sugar, or a 5-sided ring here, and it's called deoxyribose, and then you have a phosphate right there. So this is the backbone, sugar, phosphate, backbone. Then the rungs, and I'm going to show you another picture here in a little while. It's on your next page, where we put the 2 strands together. It's kind of like a ladder, where if you have, you have one side of a ladder here, and then you have another side of a ladder here, and then you have your rungs. The rungs are actually going to consist of the nitrogenous bases paired together. All right, these are your nitrogenous bases, A, T, C, and G. Adenine and guanine are similar because they're double ringed. I don't need you to write down purine and pyrimidine. Don't write that down. That's just extra information that really isn't necessary to know. I just want you to write down here that adenine pairs with thymine, guanine pairs with cytosine, and I think you already wrote that down. So now you just have the image to go along with it. But don't write down purine and pyrimidine. I'm not going to ask you about that. All right, and then you have your pairings. So we already talked about that. All right, so here is the complete picture, the structure of DNA. So here is the complete double helix. So if you look at where my cursor is, where you see blue, the blue ribbon, that is phosphate sugar. So that's the phosphate sugar backbone. This is just a really simplistic model. If you look over here though, you can see, all right, the yellow would be the phosphate, the blue would be the sugar, and then in the middle are where your nitrogenous bases are pairing up. And so what I'd like for you to know is one strand of DNA and the other strand of DNA are connected between nitrogenous bases with a hydrogen bond. So if you've taken chemistry, you know what a hydrogen bond is. If you haven't taken chemistry, that's okay. I just want you to know that a hydrogen bond is weak. It's actually a weak bond, and that's why it's represented by dotted lines. Some would say it's actually not even a bond, it's just a really strong attraction, but it is weak. And that's going to become important because when DNA gets copied, it needs to be pulled apart. So the hydrogen bonds is what allows DNA to be pulled apart. So what holds two strands of DNA together? Hydrogen bonds, and then write that they're weak. What is the shape of DNA? You're going to write double helix. All right, now I'm going to move on. All right. DNA is also considered anti-parallel, meaning that the two strands run in opposite directions. So what does that exactly mean? That means on one strand, the phosphate end is up. On the other strand, the phosphate end of the nucleotide is down. So if you look at these nucleotides, they're sitting right side up. If you look at these nucleotides, they're upside down. That just means they're anti-parallel. Phosphates are running up on this left side. Phosphates are running down on this right side. And because of that, that's what makes, because of the charge interactions, because remember, phosphates are really negatively charged, because of the charge interactions and the anti-parallel nature of how the strands run, DNA will then wrap around, forming the double helix. Right, so hopefully that made sense. So anti-parallel, they're running in opposite directions. 5' and 3', that's very confusing, especially especially if you haven't had chemistry. I wouldn't get too caught up on that for this class. I'm not going to go through that, so ignore 5', 3' stuff. I would just cross it out for now. In AP, we'll talk about what that means. It's actually pretty simple. It just means it's the 5th carbon on the sugars with the phosphates hanging off of. And then on the 3rd carbon on the sugar, you have a hydroxyl group hanging off. But some of you have not had chemistry, I don't want to overwhelm you with that. But at the point where you do take chemistry and then go into AP, we'll talk about what 5' and 3' means. But 5' just means where the phosphate group is. 3' just means where the hydroxyl group is, and they're running in opposite directions. But all you needed to get from that slide was the anti-parallel word. All right, I talked about this a little bit. I think when I talked about macromolecules or the nucleus, I know I showed you a picture of this at some point. All I want you to know from this picture is that in eukaryotes, so write that down, eukaryotes, DNA is wrapped around proteins. So in eukaryotes, DNA is wrapped around proteins. And what that does is that helps organize it and compact it into that substance called chromatin. So I think when I talked about the nucleus, said there's chromatin in the nucleus. Well, all chromatin is DNA wrapped around proteins. So if you can write that next to the picture. Okay? And then before a cell divides, chromatin condenses further to form a chromosome. You don't have to write that down, but just so you know, we take it a little step further whenever we talk about cell division. Chromatin condenses further, that's when you get an actual chromosome. All right, let's compare prokaryote DNA to eukaryote DNA. Remember, prokaryotes are like bacteria. Eukaryote are going to be your plants, animals, fungi, and protists. Prokaryote DNA and eukaryote DNA, they are both double-stranded. So if you've ever done a Venn diagram, you don't have to do the Venn diagram if you don't want to, but if you know what a Venn diagram is, Normally I would be in class and I'd be drawing this for you. It's two circles that are overlapping, and then in the middle where they both overlap, that's where you write what they both have in common. So in the very middle of the Venn diagram, you would write double-stranded, or you can just write it how it is on this slide. Prokaryote DNA is actually in a circle. Eukaryote DNA is linear. So a prokaryote has one chromosome that's a circle. Eukaryotes have multiple chromosomes and they're linear. Prokaryotes have one chromosome. Eukaryotes usually have one or more. Prokaryote DNA is found in the cytoplasm. Eukaryote DNA is found in the nucleus. And if you want to stop writing at this point, just let me go over this, and then you can pause it. Okay, because I just want to make sure you conceptualize this. I'm going to start over because I want you to really conceptualize this. They both, a lot of times students will tell me prokaryotes have single-stranded DNA because prokaryotes are just so simple. No. Anytime you have DNA, it is double-stranded. So both have double-stranded. Prokaryote DNA chromosomes are in a circle. Eukaryotes, they're linear. Prokaryotes only have one chromosome. Eukaryotes have more than one. Prokaryote DNA is found in the cytoplasm. Eukaryote DNA is found in the nucleus. And then eukaryote DNA is wrapped around proteins called histones, and prokaryotes don't have histones. And so what you would call DNA in a prokaryote, you'd say, oh, it's supercoiled. It's just kind of scrunched together in the cytoplasm. And then eukaryote DNA forms chromatin. That's DNA wrapped around proteins. So at this point, I would pause and finish writing that down, and then we're going to get into replication. All right. So I'm going to move on. So the question that we're trying to answer here is, how does DNA replicate? So replication means you're going to make more DNA from existing DNA. You're going to make more DNA from existing DNA. Who proved the semi-conservative model? Honestly, I don't even have that here. Hold on. Okay, their names are Meselson and Stahl. Obviously, that's not very important because even the biology teacher didn't have that memorized. So, I mean, I would just cross that out because I would never ask you a question about something that I didn't even have memorized myself. But in case you're wondering, Meselson and Stahl, okay, that really, props to them for knowing this and for proving this to us. But at this point, it just doesn't do anything for us to memorize names of scientists. I feel so bad about that, but y'all, I'm just being real. Good job to them. They got what they needed from that. So here's what they actually tried to figure out. And this tends to confuse some students, but I think it's really important. A is wrong. C is wrong. B is actually true. So they were trying to figure out when DNA replicates, do you end up with the original DNA still intact and then a brand new DNA? Do you end up with interspersed parent DNA with new DNA? Or do you have one strand of the parent with a new strand and then one strand of the parent DNA with a new strand? And this middle one is the one that those guys proved. It's called the semi-conservative replication model. What that means is, so here's the parent molecule, and it's called the parent molecule because it's the original. You don't actually have a parent and like a kid. Like, that's not what this is talking about. It's talking about original. I would even cross out parent and say original, but they do use parent and daughter. So you can think of parent as being original and daughter as being new. The parent molecule separates, and then each strand of the parent gets a new strand paired with it. And then you have two. So A is going to pair with T, C is going to pair with G every time. All right, so hopefully that kind of makes sense. What the semi-conservative model is, it means that each new double helix has one strand of the original and then one new strand with it. All right, and I'm not going to show a video. I'll send you that YouTube video if you want. This picture right here is showing you just how complicated DNA replication is. I mean, that's complicated. Even college students have a really tough time with this. So what I'm going to do is I'm going to list out steps for you. And you get to use these steps when you take your quiz, when you take a test, so don't stress about it. Step 1, helicase. It ends in ase, so it must be an enzyme. It unwinds DNA. unwinds it, and splits it apart. Initiation proteins separate the two strands and form what's called a replication bubble. And what I'm going to do, so what you need to do is just don't write anything down right now. Just listen to these steps. Then I'm going to go through each one. So I'm just going to give you, here's the big picture. DNA gets unwound, DNA gets separated. Another enzyme comes in and actually puts down an RNA primer. All you need to know at this point is, okay, it's RNA, it must have uracil in it. So that just gets it started off. Cool. DNA polymerase is an enzyme that comes in and starts adding new bases to a leading strand. And then you have what's called a lagging strand. So I'm going to go through that information. That is a very difficult concept for students to learn. So just know in a minute, I'm going to tell you something that's difficult to understand. Then DNA polymerase comes in and replaces that original primer from step 3. Ligase is like the glue that seals everything together. All right. So you have DNA separating, you have new DNA being put down, and then basically you have something sealing it all together. That's the gist of all of this. So let me go through each step. Now you're going to write. Step 1, helicase unwinds DNA at the origin of replication and creates replication forks. So pause to write that down. Then I'm going to talk. Now I'm going to talk. Here's your original DNA right here. So see where my cursor is? You have 2 strands of DNA. Helicase comes in at multiple points. So you have a long double helix. Helicase comes in at multiple points and opens it up. It's like if you had like, I don't know, it makes me think of if you have like a superhero or something like the Hulk, or I don't even know, if they're in jail and they have bars in front of them and they grab the 2 bars and they pull them apart, and that's how they escape from jail. That's just what I think of what helicase does. It pulls DNA apart to form these little bubbles, and then the light blue is showing you the new DNA that is being matched up to the old DNA. And so DNA is being replicated in both directions, to the left and to the right inside that bubble. And this is a very, very efficient process. And then at the end, now you have 2 new complete DNA molecules. So if you follow this picture down, you see helicase is pulling DNA apart, new DNA is being made, and then you have $2 molecules. So these are called replication bubbles. So I'm sorry that step 2 wasn't actually written on here. I would say step 1 is helicase unwinds DNA. Step 2, it's creating replication forks. So unwinding is step 1, replication forks is step 2. Replication, the fork part is where the arrows are. The bubble is the whole thing, in case you were confused on that. All right, now we can go on to step 3. All right, so step 3, primase is an enzyme, and it's pink right here. It adds an RNA primer. So the way I think about this, so anytime you see blue, it's DNA. Anytime you see red, it's RNA. We like to color code things in this class. So this is the way I think of it. The enzyme that actually comes in and adds new DNA and like makes the new DNA and puts it all there needs some encouragement. Kind of like, let's say celebrities are going to an event. What do you do before the celebrities actually enter the event? You roll out a red carpet for them to walk down, and that encourages them to go to the event because the paparazzi is taking pictures and it's amazing and they're getting a lot of attention. This is the way I think of primase and RNA primer. Primase is the guy that's coming in and laying out a red carpet, trying to encourage DNA polymerase to come in and start adding the new DNA, right? It's like an attraction. So RNA primer is attracting DNA polymerase. Then DNA polymerase sees that RNA primer is like, all right, I'm going to do this. That's encouraging. And he comes in, they don't have genders, obviously. It comes in and starts adding nucleotides, new nucleotides. Don't get caught up on this phosphate thing. All I'm trying to convey to you in this picture is this takes energy. We need the cell needs ATP to do this. So how does DNA replication relate to cellular respiration? Because the ATP made in cellular respiration is used in DNA replication. It requires energy. So that's all I'm trying to convey. Don't get caught up in this picture. DNA polymerase comes in and starts adding to the new strand. Okay? DNA polymerase comes in and starts adding to the new strand. All right. So remember, every time you see dark blue, that's the original strand of DNA. Every time you see light blue, that's the new DNA. And every time you see red, those are your primers. This is what the difficult concept that I was talking about earlier. And the concept is leading strand versus lagging strand. DNA polymerase can only add in one direction. So on this top one, it can only add going, and this is the origin right here. This is where helicase unwound everything. It can only add moving left towards the opening. Down here, it can only add moving right towards the opening. And it starts right here. So you have a leading strand that's continuously being synthesized towards the replication fork. Now I'm sure I'm starting to lose some of you already. And then you have a backtracking down here. There's a backtracking. Those are called the lagging strands. So is it absolutely necessary that you understand leading and lagging strand for just a regular cell bio class? In public schools, you actually do. Freshman biology, they do have to know leading strand, lagging strand, and Okazaki fragments. For this class, though, I think you can get away with not completely understanding it. In AP, though, we're going to go back over it, and you really will need to understand it. Those that are moving on and using this as a prereq. It has a lot to do with the 5' end and the 3' end. DNA polymerase cannot add a new nucleotide to the phosphate end. It can only add to where the hydroxyl is. Therefore, at this point, it can only move left right here, it can only move right here, and then it has to backtrack. This is called the leading strand. This is called the lagging strand. The lagging strand is also known as Okazaki fragments, and they're the fragments that are backtracking, that are backtracked basically. And they're called Okazaki fragments. And what happens is DNA polymerase 1 comes in and replaces all that red with light blue, and then ligase seals them all together to form one continuous strand. Here are and then you have a picture that is summarizing everything that I talked about with replication. This is difficult. This has a lot of steps. Okazaki fragments, leading strand, lagging strand is something that I honestly did not fully understand until my second or third year of college. It is something really difficult to understand. So if you understand it, I mean, good for you at this point. If you don't, that's okay. I just need you to know that there is a leading strand and there is a lagging strand that consists of little fragments at this point. All right, here's a list of enzymes and what they do to kind of summarize what we talked about here. Helicase unwinds, single strand binding proteins are just going to hold it. They're like the hands that hold DNA apart. Topoisomerase is going to relieve strain upstream. So remember, DNA is coiled around each other. Topoisomerase is just going to come right here, and it's an enzyme that just kind of relieves the tension as the new DNA is being made, and as DNA is getting pulled apart, moving in this direction. It just relieves the tension. I'm not going to ask you about topoisomerase, but it's just here for completion. Primase, remember, is going to lay down the red carpet. DNA polymerase is going to be responsible for the leading and lagging strands. DNA polymerase 1 is then going to replace all of the primer with DNA, and then ligase seals it all together. Here's the beautiful picture, summary picture. I would just, take a look at this and try to go back to the steps that you wrote down. Go back to the steps you wrote down, and I would number on this picture what step correlates to what part of the picture. That's what I would do. Okay? And then just do your best to understand it and move on. And then please write down any questions you have, like a question mark just next to something. And in the Zoom call, if you feel comfortable, or at the end of the Zoom call, you can wait to take your quiz. You don't have to take your quiz before the Zoom call. At the end of the Zoom call, stick around if you don't want to ask in front of everyone, or you can hang up from the Zoom call and then email me and say, hey, I really want to go over these DNA notes because I'm really confused. I will help you with this. All right, last page, proofreading and repair. So DNA needs to be proofread because if mistakes are made, that will lead to a mutation, which will lead to a disease. So DNA polymerase is going to proofread as it goes. It's kind of like if you're on the computer and you were typing and you realized, oof, I just put the wrong letter, and you put a backspace or a delete and you wrote the right letter. That's what DNA polymerase does, is it proofreads and it corrects itself as it goes. But some mistakes, I don't know why I emphasize but so much. It's like DNA polymerase is really trying, but kind of like humans, like me, especially Teams. Teams does not like tell me when I've accidentally hit the wrong button and spell something wrong. So just so y'all know, I know how to spell. It's just my nails kind of make it so I put the wrong letter sometimes, and Teams doesn't tell me. So just like DNA polymerase, I make some mistakes sometimes that I miss. So DNA polymerase is going to miss some mistakes too. About 1 in 100,000 get past this initial check. That's kind of a big deal. That's a lot. So what we need are some special enzymes that come in and repair mistakes. So DNA polymerase proofreads as it goes, but then these things called nucleases come in, cut out mistakes or damage, and then DNA polymerase comes back and ligase and they fill in the gaps. So DNA polymerase is like you writing a paper. You turn it in and your teacher is like the nuclease that like puts red marks on everything, and then you come back, you and ligase come back and you correct what the teacher told you to correct. So ultimately, after all of this is done, you end up with, I'm trying to get to the point here. I put a bunch of extra information, but the next thing that you need to say is the errors. All you need to write down is, after all of that's done, it's a 1 in 10 billion. 1 in 10 billion. So that means maybe a human will have one base pair that's wrong. Maybe. And it could even be silent. Humans have about 3 billion nucleotides. So if the mistakes are 1 in 10 billion and you have 3 billion nucleotides, there's a chance that you don't even have any mistakes, right? And that's a good thing. That's a really good thing. Okay. So last thing that I want to talk about is we have linear chromosomes. Every time DNA gets replicated, the chromosomes get shorter. So I want to talk a little bit about why. At the very end, so let's say this is the very end of a chromosome, that last primer right there is unable to be replaced. And it has to do with DNA polymerase only being able to add to the hydroxyl or 3' end. So DNA polymerase, and if you don't understand 3', 5', that's okay. Just take my word for it. DNA polymerase cannot replace the very end primer on a chromosome. So that's what you would write down. Why does DNA grow shorter and shorter with every replication? Because DNA polymerase cannot replace the primer that's at the very tip of the chromosome. Therefore, it ends up getting chopped off. So that would be very unfortunate if that was actually DNA that like coded for something. If the ends of your chromosomes the DNA that actually coded for something really important was getting chopped off every time a cell divided, you probably wouldn't survive the first division. So we have a little safety net in place, and these are called telomeres. Telomeres are repeated units of short nucleotide sequences at the very tips of our chromosomes. So it's basically junk DNA. It's garbage DNA. I mean, it's not junk or garbage. That's offensive. It's very necessary. But when I say junk and garbage, I mean it doesn't code for anything, but it is very, very necessary. When you think of telomeres, I want you to think of the aglets at the end of your shoelaces. Every time I say the word aglet in class, people they say Phineas and Ferb. I don't even know. I mean, I've been teaching this class for like 8 years now. I don't even know if kids y'all's age even watch Phineas and Ferb. My son did though. So anyways, aglets. Everyone knows from that episode what an aglet is. The telomeres are like the aglets. It keeps your shoelaces from fraying, right? And you could cut a piece of the aglet. Like let's say you cut a millimeter, then you cut another millimeter, then you cut another millimeter. You'd be fine for a little while, right? But at some point in time, that plastic's going to you're going to run out of it, and then you're going to end up fraying your actual shoelace. This is exactly what happens with DNA replication. Every replication, it gets cut shorter and shorter and shorter, but we have telomeres to protect, that non-coding DNA to protect, but at some point, after a certain division, you will reach a point where, or the cell will reach a point where it runs out of telomeres and it starts cutting into coding DNA. And at that point in time, what I want you to write down is, when a cell runs out of telomeres at the tips of its chromosomes, at that point in time, it will undergo apoptosis, also known as cell suicide. I hate saying that, especially this day and age. It's kind of a really sensitive word, but you actually will find that term in a biology book. Apoptosis, quotations, cell suicide. And this is where lysozymes are triggered to release hydrolytic enzymes, and the hydrolytic enzymes are going to chop up the entire cell. So that's pretty cool. That's great. Because if a cell gets old enough, you don't really want it like functioning. Kind of like when a person gets old enough, you don't want them driving a car anymore. You got to take their driver's license away. It's the same thing. If a cell gets too old, you don't want it to function anymore. In cancer, one of the reasons why cancer cells divide out of control is that there's an enzyme that is present, and this enzyme is called telomerase. We don't want telomerase in our cells. The only place we want telomerase is in germ cells, and those are the cells that are going to divide to make sperm and eggs, and we're not going to talk about, yeah, we actually will talk about sperm and eggs in this class. Anyways, germ cells are the cells that divide to make sperm and eggs. You want telomerase working in them to replace telomeres when cells divide, to replace that DNA. But in any other cell in our body, we don't want telomerase. But what scientists are finding is that in cancer cells, telomerase is an enzyme that comes back and adds the telomere back on. So when we talk about cell division and we talk about cancer, I want you to remember this, because cancer cells are considered immortal. They never die. And the reason why they never die is because telomerase comes in and keeps adding telomeres back on. Telomeres are a good thing. Telomerase is not such a good thing. Cancer is not such a good thing. And so just so you know, this is going to kind of lead into a discussion about cancer. And so here's a visual for you. So telomerase is an enzyme that adds telomeres back on, is all I'm trying to show you in this picture. All right, and that is it for this slideshow. Please let me know if you have any questions on anything that we went over, or that I went over and that you wrote notes on. And like I said, you can do it through email, through Zoom, or anything. I want to help. This is kind of a difficult topic to cover in such a short period of time. And just so everyone knows, I've probably said this before, but each day in this class represents an entire week. So this slideshow, I would have covered in an entire week in a cell bio class. So it's difficult, but everyone's doing great. Let me know if you have any questions, and I'll talk to you guys later.